glucose. Everybody's cool with that?
Yeah.
Glucose, can glucose get through the cell membrane?
No.
No, it's a large molecule. So the glucose has to go through a receptor, okay, or go through a channel, and it has to do something known as facilitated diffusion. Is that term, you guys heard that before? Facilitated diffusion?
Active transport.
Huh?
Is that the same thing as active?
No. Active transport is going against the gradient. Diffusion is going with the gradient. So what happens is, glucose wants to get into the cell, right? But it can't. Something has to facilitate the movement of glucose into the cell. And what facilitates the movement of glucose? Insulin. Yes. So insulin facilitates the movement of glucose into the cell. Once the glucose enters, okay, right here at the cell membrane, inside of the cell, our first cycle occurs. Our first cycle occurs, and I'm just going to draw as kind of a circle. All right? What is that first cycle that happens?
Breaking down glucose.
Okay, yes, glucose comes in, okay? Glucose comes in, and it's broken down into pyruvate. Okay, it's broken down into pyruvate. What is this cycle called?
Glucose cycle?
What? Very close. Glycolysis. This is glycolysis. Now, do I produce any ATP in glycolysis?
2.
Yeah, I do. I produce 4 ATP, but I end up using 2 to actually run the dang thing. So I only have a net, at the end of the day, I only net 2 ATP. Is everybody cool with that? Not a whole hell of a lot of ATP, right? Not much. Do I need oxygen for glycolysis to work?
Yes. Yes.
No.
No. Glycolysis is the anaerobic part of respiration. Okay? Glycolysis is 100% anaerobic. What does anaerobic mean?
Without oxygen.
Awesome. So when we talk about going into anaerobic metabolism or anaerobic respiration, this is what we mean. This is the only cycle that's working here. Is everybody okay with that? With glycolysis? Okay? Okay, cool. Now, the pyruvate then moves to the mitochondria. Okay? Pyruvate comes into the mitochondria. So I'm going to just kind of draw a really, really large mitochondria here, just so I can show you. And there are 2 cycles that occur within the mitochondria. Okay? So the first cycle, draw here. The first cycle is known as the Krebs cycle. The Krebs cycle. Some people call this the citric acid cycle. Some people call this the carboxylic acid cycle. And we know that, from yesterday, we know that citric acid and acetic acid, those are examples of carboxylic acids, the COOH group. Okay. So pyruvate's going to come into the Krebs cycle. What's going to happen is, pyruvate is glucose, it's a modified glucose, and so you have this carbon chain, right? You have a little chain of carbons, and then off of that chain of carbons you have all these hydrogens, right? So you have something kind of like this, right? Hydrogens and OHs, just like we talked about yesterday. Something like that, okay? I don't know if that's glucose, but something like that. So what's going to happen is, we're going to start tearing these hydrogens here. We're going to tear these hydrogens off of that pyruvate. And we're going to tear those hydrogens, and those hydrogens are going to be transported to another cycle within the mitochondria, okay? And there are transport molecules, NAD and FAD, okay? And the NAD actually load the hydrogens on, so they become NADH. plus, I believe, something like that. And then these molecules then dump those hydrogens into another cycle. I'll get to that in a minute. Okay. So as I'm tearing hydrogens off, what do I have left over here? Carbon and oxygens, right? Carbons and oxygens, pretty much, because I'm tearing the hydrogens away. So if I have lots of carbons and oxygens floating around, what do you think I could make? CO2, right? Yeah. All right. So the carbon dioxide waste from cell respiration comes out of the Krebs cycle. Okay. CO2 is produced as a waste product in the Krebs cycle. Everybody okay with that? Yes, sir.
The NAD and then it's FAD, right? And
Yeah,
then it
NAD.
goes to NAD, is that H?
Yeah, NAD, pH, and FADH. It's something like that. I don't really know the exact. I don't want to say that's the exact thing, but they load up the hydrogens. The hydrogens are loaded onto the NADs and the FADs, and then they transport the hydrogens over here.
And what is that cycle? That second cycle?
The second cycle?
The other one.
Yeah, haven't gotten to that one.
Oh, okay.
Yeah, I want to make sure. Are we okay with the glycolysis? Are we okay with the Krebs cycle? Okay. So now these hydrogens go over to here, and this is known as the electron transport system. Some people will also call this oxidative phosphorylation, because I'm phosphorylating ADP to make ATP, and oxygen might just be involved as well. Okay, so what I'm going to do is I'm going to enlarge this here. I'm going to enlarge the Krebs cycle, or the electron transport. And so what we have is we have a membrane, the membrane of the mitochondria. And then I have several proteins in that membrane. All right, several proteins in that membrane. Okay, and the NAD and the FAD are going to dump those hydrogens off over here. And the hydrogens will be ionized when they get dumped off. So they will actually be H pluses. So what are they missing?
An electron?
They're missing electrons, right? So we also have electrons that are getting dumped off. Now what happens is the electrons have energy, right? They have energy, right? They have kinetic energy, they're moving. The electrons are going to move through these proteins, okay? These proteins are embedded in the membrane, and the electrons will be used to power these proteins. Does that make sense? Kind of provide some power. And then what happens is hydrogen ions build up. And I have a whole bunch of hydrogen ions here. Where do these hydrogen ions really want to go? They want to go over here, right? Because diffusion, remember, diffusion is the movement from high concentration to low concentration. They want to move through here, they just can't. Is everybody cool with that? Now what happens is at the very end of this chain, there is a special protein. Okay? Special protein called ATP synthase. ATP synthase. All right? And what's going to happen is this is a channel, it's a little channel. All these hydrogens that have been built up are going to flow through this channel and they're going to come out here. Is everybody cool with that? They're going to flow through this channel and they're going to come out here. And as they flow through, it's kind of like, imagine like water going through a turbine, like a hydroelectric plant, and the water, you have a dam. And imagine this is kind of an opening in the dam and the water's flowing through there and the water can turn a turbine. Well, these hydrogen ions are going to turn, this enzyme, this protein, literally turns as those hydrogens are flowing through. And what it does is it can take an ADP molecule as it turns and add a phosphate to it and then spit out ATP, and then it turns around, grabs another one. and spits them out. Does that make sense?
Can you say that one more time?
Okay. So,
So it's from, go ahead.
so imagine that these hydrogen ions are like water, okay? Imagine that this is a reservoir here, okay? This is a reservoir of hydrogen ions, and they're flowing through the dam here. Imagine this is a dam. They're flowing through the dam, and imagine that this is a turbine, okay? Which it is in a way. It's a turbine, and as those hydrogen ions are flowing through, ATP synthase starts spinning around. Is everybody okay with that? Now, that action, that movement of the ATP synthase, what it does is you have a molecule of ADP here, adenosine diphosphate, okay? Adenosine with 2 phosphates, and I have a phosphate here. Can you guys imagine that? And as that thing moves around, it can combine them and spit out ATP. And then it just keeps moving around and spitting out ATP as it spins around.
Where does it grab that other phosphate from?
The phosphates are coming from other cycles that we're not really going to talk
And the
about.
ATP, where was that at and how did it get there?
Yeah, yet another cycle.
Right on.
Yeah.
Good.